In order for microbial oceanographers to understand the diversity of marine microbes, they must employ a technique to look at the blueprint of life, DNA. Today I followed along with the diversity group as part of the Seymour summer course.
I'm Emily Fake, I'm from Griffith University in Australia. Every living organism contains DNA, and DNA is like the instruction manual for how a cell is going to function. And how we use it is that DNA sequences differ between species. So we can look at the different sequences and determine how many different species are within a sample, and then also maybe perhaps how they're functioning.
And one of the first things we do with the diversity group is we have to collect the microorganisms in order to be able to extract their DNA to do the analyses. So in order to collect them, first we send Niskin bottles down in the water, we bring the water back on deck. We collect the water in bottles and bring it back to the lab. Then we hook those bottles up through a pump and push the water through a filter with very small pore sizes. The pore sizes are so small that all the microorganisms get trapped right on the surface of the filter. And then we can take those filters and extract the DNA off of all the microorganisms. I'm here with Paula Moraes from Brazil, and she is showing one of the filters that we've just completed filtering. And you can see it's changed color a little bit. That green coloration there is from all the phytoplankton that have been trapped on the surface of the filter. And now we're going to take this filter and put it into a tube and extract the DNA.
So I'm Ramiro, I'm from Barcelona. We got DNA in this little tube today, and we want to sequence this DNA and to know what it is. But this is not enough DNA for going directly to sequence it, so we need to amplify this DNA, which means we need many copies of this DNA for continuing working. So for doing that, we will use PCR, which is putting a little bit of DNA in the small tubes, putting them into this machine and incubate for about 3 hours, and then we will get millions and millions of copies of the DNA that we want.
Okay, so this is the most exciting part of the experiment, because now we're going to check if the amplification of the DNA worked out. We put the DNA on the gel, an electric current is applied, and according to the size the DNA is separated. Additionally, we put a fluorescent dye, so in the end we're going to see is there enough DNA according to the intensity, and do we have the right size of the DNA so we can continue working.
The last step in this process is to take these samples back to shore to be sequenced. Another day at sea comes to an end. Until next time.